Dyra is the first student who got the feels. Like I got the feels when she said that I influenced her path. So this is the first time I've had that experience. So thank you, Dyra. I'm gonna, as Dyra mentioned, I've recently moved to UCLA from OHSU. I'm gonna talk fast because I really had a lot that I wanted to pack in here. And I'm gonna tell you about work we've been doing in our large consortium projects like Cancer Genome Atlas, and then talk about what moved me to UCLA in terms of implementation science and genetics very broadly. So I'm going to start with these two big projects, the Cancer Genome Atlas and the Pan-Cancer Analysis Working Group projects that analyzed ICGC and TCGA data. And I'm going to do it specifically in the context of tumor evolution primarily. And so many of you are probably familiar with the Cancer Genome Atlas that really used a very large amount of ARA funding to complete an enormous genetic profiling of a very large number of tumor types, nearly 50 tumor types, more than 20 tumor types over the 10-year span of its lifetime. And as a member of the TCGA from the founding, I was there for most of this. And for those of you who would want to give credit to someone, you should actually give the most credit to Kenneth Shaw at MD Anderson, who made sure we actually had samples, because that turned out to be the single biggest problem. We had lots of people who knew how to do genomics. We had lots of people who knew how to do informatics. We had no one who knew how to get large numbers of samples systematically at very high quality. And so Kenneth Shaw is often underappreciated, and I would highlight her. So I want to start with this concept of tumor evolution, and many of you will recognize this model from Vogelstein and Kinsler on how colorectal tumors develop, where you have a progressive series going from normal epithelium to metastatic disease. And they proposed a series of acquisition of oncogenic lesions that progressed in some ordered way, right? That they suggested that the first things that happened were APC lesions, then typically KRAS lesions, and then alterations in SMAD signaling, and then finally loss of chromosome regulation in P53, leading to these carcinomas, and then finally other changes leading to metastatic disease. And one of the big problems for most tumor types is you don't get all of these samples, right? In colorectal tumors, because of colonoscopies and other vision systems, you can go in and clip out early adenomas. You can go and get these intermediate adenomas. You can get the late carcinomas, and you can profile them and see what's going on in them. But for the vast majority of human malignancies, we don't have that opportunity. We get this frank invasive disease or metastatic disease of presentation. And so the screening process is not coupled to the removal process in the same way. And so getting these kinds of ordered samples where you really believe the progression series is correct is usually impossible for most human malignancies. And that's led us to models like this one, which suggests that clonal expansions arise, leading to the acquisition of these stars here that are driving oncogenic events that lead to clonal expansions. And the most recent common ancestor would be the set of genetic events that have accumulated in the tumor that are present in essentially every cell of an evolved tumor, but that there could be subclones that frequently arise later on and begin to take over, potentially interacting with each other in complicated ways. And so when we think of a tumor, we're really thinking of a complex ecosystem that has evolved under selective pressure for decades. So the PCOG project had many specific arms that all interacted heavily with one another. PCOG Evolution and Heterogeneity, led by myself, David Wedge, and Peter Van Loo, was responsible for cataloging evolution heterogeneity within the 2,658 whole genomes of PCOG. And specifically, we wanted to build out maps of tumor evolution using what was available to us, which is point estimates at the end of surgical resection, right? We got a piece of tissue, and that's all we had. We didn't have time series back in time. We didn't have any retrospective way to get these. And so we wanted to understand how we got to these evolved tumors from what we were given at the end. And I don't see Ludmil, but we were heavily influenced by our colleague here, Ludmil Alexandrov, on the use of mutation signatures, and look at those as well in the context of tumor evolution. So the way we do this is built off of, started with a postdoc in my group almost 15 years ago, who we realized that if we looked at tumors at the end of evolution, they would show all the marks that they had acquired during the process of tumor evolution. And that's normally not terribly informative because you say, okay, they have all these things. But we realized that there were specific things that could happen that would be informative. And that was anytime you duplicated a chromosome, a mutation that occurred before that duplication is on both copies of the duplicated chromosome. But mutations that happen after the duplication are only on one copy of a chromosome. And so you can implicitly order things that happen before or after a duplicated chromosomal event by whether or not a genetic variant is estimated to occur on one or two or multiple copies of a chromosome, on those chromosomes that are duplicated. And so you can infer when this chromosome is duplicated and infer which variants were before the duplication, which ones were after, on any duplicated chromosome. And so you can use this to actually get some quantitative estimate of when during tumor mutation time, a genetic set of events has occurred. And this is what we call the 2+1 state, where one chromosome has an extra copy. There are two chromosomes of one of the parental alleles and one copy of the other. And so I'll use this nomenclature, 1+1 for what we're all born with typically, 2+1 for a duplication, 2+2 could either be gain of both chromosomes or it could be indicative of a whole genome duplication. And so that framework, n+n, is how we're going to keep track of our copies of chromosomes. So using that, you can do some very simple things like, for mutations in gene X, is it likely to be early or late, right? Based on this timing mechanism. When it's informative, can I determine whether or not it happened before or after duplication? Many tumors have whole genome duplications, and given that whole genome duplications are prevalent, you can minimally get a big benchmark on, is it before or after a WDD event? In this case, you can see that P53 is almost always an early lesion in the cancers in PCOG, the 2,658 tumors in PCOG. It's almost always early. KRAS, almost always early. PIK3CA, almost always early. So the common cancer drivers that many of you will be familiar with are almost always early. And we can segment between truly early, like during mutation time acquisition, and at the junction between clonal and subclonal, right? So sometimes we have subclonal mutations that are only on less than one copy of a chromosome. And so this figure just shows the specific genes that you may be familiar with and whether or not they're estimated to be early and whether or not they're estimated to be clonal. So those are two sort of separate times in tumor evolution. If you look at P53 specifically, you can ask in different tissue types, is it found to be early or clonal? And you see here that in pancreatic, it is almost certain that P53 is one of the earliest events that's happening in pancreatic cancer. The same for ovarian cancer. In fact, we had a separate paper we published before this showing that in high-grade serous ovarian cancer, the P53 mutation is almost certainly the first event that really drives most of these tumors. And so you can then estimate whether or not where in the hierarchy of order and necessity in any individual driving oncogenic event has happened. And to quantify what I sort of briefly described before, which is what driving events are common, what we see is that if we look at the clonal early period, those genetic events that happen at the very earliest, they are almost always in a key set of core genes that you're familiar with, KRAS, P53, PIK3CA, APC. These core oncogene drivers are almost always in that earliest bucket. You only need just less than 10 genes to identify nearly half of all of the early drivers in these tumors. Whereas if you go later in the period, a larger swath of genes open up to tumors that allow them to evolve and adapt in the spaces that they live, such that you get a very long tail of genes, a wide variety of genes that show up later, and that expands further and further into oncogenesis. And you can commonly see this in many tumor types where you might see that a gene like PBRM1 in clear cell kidney cancer is an important known oncogenic gene, but it's only found to be mutated in a very small fraction of cases because there's multiple different paths through the node that might involve PBRM1. You might use SETD2 instead, for example. So what Moritz Gerstung and Ignati Lesher did was figure out a way to actually build this into a model rather than just make these qualitative statements. They actually figured out that you could attempt to reconstruct the likely order of events using competition models that are very similar to the sports statistics you use to figure out which sports teams are better. And you could compete different possible paths against each other and then put them in order. And so here's a hypothetical set of 100 genomes that have evolved with a set of different oncogenic drivers at different frequencies. And they're fed in order. And then from that, you can reconstruct out on the right what the likely order of events was, right? So given some known order from a small set that's incomplete, you can output the likely priors of what happens before what. And so here's actual data from individual tumors where we have real statements we can make. So the 3 specific tumors, a colorectal tumor, a pancreatic tumor, a GBM, we can say that for the colorectal tumor, there was an APC mutation, a KRAS mutation, and a SOX9 mutation, then a series of chromosome losses. And those all happened very early. We can't order them against each other, but we know that they're before the whole genome duplication, right? And then after that, there was a loss of P53, and then there was a subclonal loss in APC, right? So we can put those things in order and say, this is an order that's happened in this tumor as a piece of information. There's a pancreatic endocrine tumor, has very little information. It has some things that we can't distinguish against each other, like the top branch and the bottom branch. We don't know how they relate to each other against each other, but we know that they're both before the subclonal events that happen. And then the GBM has its own behavior. And so if you do this and you feed in the actual data from hundreds of tumors into a model like this, and then compare it back to the Kinsler and Vogelstein diagram for colorectal cancer, we recapitulate that extremely well. We think the earliest event is APC. We think the next earliest event is KRAS. We think when PIK3CA is present, it's also extremely early. We actually think that P53 is earlier than this MADS, and I think there's a real question there, because when you have these serial progression models, you are only observing what progressed, right? But you're making the assumption that the order and acquisition of the things you pull out of someone is actually correct. And it may very well be that the acquisition of SMAD mutations blocks tumor development from progressing further. And so those tumors are actually stuck there and they can't go further and they need the P53 to happen beforehand. So I actually prefer our way of doing it because we see what's coming at the end, but proving this is obviously tricky when you have 2 competing models. So we've reconstructed this in this paper from the Peacock paper for pancreatic and as well as GBM, and have similar models for all these. In fact, all the tumor types in Peacock have these underlying models that identify early events. Something that was particularly interesting to us is that in the GBM tumors, there are a set of chromosome gains. They happen ridiculously early. We estimate they happen before the age of 10 in nearly every single one of those individuals. 3 specific chromosomal gains that are very high prevalence and happen over and over and over again very early. They're amongst the earliest events. So the next thing I want to talk about, hopefully many of you have seen this because of Ludmila's beautiful work, the process of taking these big panels of mutations and turning them into mutation signatures. And I'm not going to walk you through it, neither because I have time, nor can I actually explain the math properly to you. But Ludmila can, and you can go ask him about it. But the idea is we're going to turn sets of thousands of mutations that we get in these tumor genomes into specific mutagenic signatures that are indicative of some biological process or mutagenic process that has occurred. So this is the signature for UV light, signature 7. It has a very characteristic substitution signature where C's change to T's in a very specific set of contexts preferentially. And using that, you can then take that and intersect that with our mutation timing estimates. So we think this is an early mutation. That mutation is likely caused by mutagenic process X. Therefore, mutagenic process X happened early. Or that mutation happened late, and we don't see that mutagenic process happening early. So we can distinguish this, and here's an example of a specific tumor in which the UV mutagenic signature was highly prevalent early in tumorigenesis, and then is taken over by one of these unknown signatures at the time, SBS38, right? So the tumor has changed its the underlying processes that are mutagenizing the genome over time, and we can now distinguish this and separate when things happen in terms of changes in the DNA sequence as a function of time. So the PCOG paper completed with a model like this for each of the tumor types with the set of oncogenic drivers that happened during the early development period, the time that we expect whole genome duplication to occur, a set of common drivers that happen afterwards, the time from the most recent common ancestor. And one thing I didn't show you is that using the clock mutations from the timing mutagenic spectrum work, you can actually back infer some of this to actual chronological time, assuming that the clock mutations really occur in something like real time. And so that allows us to put timestamps on things like we think this happened at age 3, right? So you can sort of make these qualitative statements that turn into quantitative statements. So moving forward, this is work from a postdoc of mine in the lab now, Toby Baker, and continuing work he did with Peter Van Loo when Peter was at the Crick, which is to push this even further. And so the idea that Toby and Peter had was to see if we can even extract more information from these statements we're making. And the first thing they did was copy number states in tumors are actually incredibly complicated, right? Like there are copy number gains and losses, but most tumors are not diploid. The average tumor that kills someone is something like tetraploid or triploid. How did it get there? And what paths did it use to get there? And using a bunch of techniques that have been developed previously, we can often identify tumors that have undergone whole genome duplication, going from 2N to 4N. But it's often difficult to infer how you get to some specific copy number state on some specific chromosome. And if you look in, I believe this is the Hartwig samples. If you look in the Hartwig samples, you'll see that there are a large number of tumors that don't have whole genome duplication, but an equally large number that do have whole genome duplication. And if you look at that N+1 state framing that I used before, you can see that diploid 1+1 is a fairly common mode, right? There are tumors that are basically diploid. But you can also see that 2+1 is a very common state to be in as well. Lots of chromosomes exist as 2 copies of one of the chromosomes and 1 as another. And I want to highlight this 3+2 state. So that's 3 copies of one of the paternal chromosomes and 2 copies of the other. And the question is, how do you get there? So you could go through a process where you had your maternal and paternal alleles, you gained one of those chromosomes, then you had a whole genome duplication, and then you lost one of those chromosomes, right? That's one possible route to a 3+2 state. The other possible is you started with maternal and paternal, you'd had a whole genome duplication, and then you gained one of those chromosomes, right? And so can you distinguish those, and do they tell you anything interesting? And Toby realized that although this is the most parsimonious route, right? It only requires 2 steps rather than 3. But Toby realized you can actually keep track of these things. And by keeping track of the number of mutations that are on 1, 2, or 3, or 4 copies of a chromosome, you can infer when these events might have happened. And so if we draw this pathway, the WGD pathway, gain WGD pathway, the paternal allele underwent a gain event, and it split into 2. And each of those duplicated chromosomes split into 4, and then the red was one that was lost. So you end up with 3 gray chromosomes in the left-hand panel on the right. Whereas the maternal allele underwent the WGD and now has 2, right? So there's 3 gray paternal alleles and 2 gray maternal alleles, the 3+2 state. Whereas this version has a different process. And so you have different expectations on the number of mutations that are on 1, 2, or 3 copies of the chromosome, depending on which of the routes that it got there with. There are a lot of such paths. Toby actually iterated them all out. This is the paths that are available to a 4+2 state. I took out the slide showing all the paths to like the 5+5 states. But the key is that the number of mutations on 4 copies is proportional to the timing of the first gain, and the number of mutations on 2 copies is proportional to the second gain. And so with that, you can then take any given tumor that has a 4+2 state and say, are the data more consistent with the most parsimonious route or one of the more complicated routes that you might choose? And you can see that the data are sometimes are more consistent with a path that is not the most parsimonious, right? Nearly a third of the time, the way you get to in this case, the 4+2 state is not the straight gain whole genome duplication. It's something else as inferred by the underlying data. And then Toby got even smarter. He said, wait a second, we can do something even more clever. We've been timing our gains based on our SNVs. We can time our SNVs based on our gains and go backward, right? And so now we can say, based on this process, where did these specific mutations happen? And we can do even better fine-scale timing driving, driver timing. And so for a mutation that's present in the earliest state, it's in this window. For a mutation that's present only on one chromosome, it's in the late window. And then if it's on one of these two, it's in this middle window. And we have a trick that we're going to try to get to in the next, that I'm not going to talk about, using long reads where we phase everything, where you can then now define whether or not it's maternal or paternal, and then you can then winnow this down to either the early or late phase. So we're going to have a set of processes where we can put these mutations in very fine-grained windows based on this step. And then any mutation that's subclonal can be classified that way. So then you can look and say breast cancer and say for PIK3CA, that's almost always early. P53, that's also almost always early. The mutations that occur in BRIP, they occur later in this process, and sometimes you have these mutations at PAT4 that I know nothing about. And they happen subclonally. And so you again get our diagram of ordering like this. And again, we can get our aggregate driver ordering, as I showed you before, for this model. And so in this case, P53 is early, APC, and then TERT and KRAS, et cetera. Okay, so now we can get even fancier. I showed you how we could time mutation signatures. Anytime we have a chromosome gain that we've estimated at a specific time, we can take the mutations that are on that chromosome and infer what mutagenic processes were active on that chromosome, and then time when the mutagenic processes were happening in finer grain detail. So given some chromosome that duplicated very early, if there are no mutations of signature X present on that chromosome in that early window, we can infer that signature X was not active at that time, even though it was active at some later time. And so we can deconvolve these very specific mutagenic signatures into very fine-grained timescales. And so we're in the process now of recomputing all the data to really tease apart when all these mutagenic processes might happen. And so Toby currently is doing this in sort of this classification-based way rather than a kinetic model where you've just got classes of stable, early, late, peak, trough, and other. And the reason for that is we have relatively few copy number gains, and so we don't have as much fine-grained discrimination as we'd like. But that can lead us to statements like carcinogens are early, right? And that we can infer that nearly all the carcinogenic activities present in these data sets happened early in tumor genesis. The treatment-based ones, as you might expect, they happen when people are being treated with a drug, and they happen late, so that's good. Age tends to happen early, but some of them are not uniform. Endogenous processes, for example, DNA mismatch repair, often kick in later in the process. They're not necessarily the first thing to go. The ApoBec enzymes that are involved in viral detection and host defense against viruses, they almost always are in the later period. And then we have a set of etiologies, mutation signature etiologies we don't understand. And again, these can happen at any time. And so understanding when they're active might help us figure out what they're caused by. Okay. So altogether, our evolution modeling can help us put the chronology of cancer development in order to both better understand detection, drug sensitivity, various other things. We can continue to push this and observe these signatures and exposures potentially. And as we continue to add more and more cases, and this whole genome sequencing is dropping, I got an email the other day from a colleague, $80 genomes are here, right? So like, you know, as we get cheaper and cheaper, more whole genomes, if we can get them into the research enterprise from the clinical enterprise, we'll have continually more and more data to build these kinds of models off of. And ideally, we'd start being able to compete them against each other in very specific cases where this set of tumors has these set of mutations. They have a different order than in this other case, or when the mutagenic process X is active, has that changed the timing of other things? And so ideally, this is a data-driven enterprise, and more data will give us better answers. Okay. And we're super excited about the long range sequencers. They're not cheap, unfortunately, but they do allow us to put on methylation and other types of information and phase things better, as well as do structural abnormalities. So we're investing very heavily in that. Okay. I want to do a little advertisement for a project that's part of the GDAN, the Genome Data Analysis Network. This is a follow-on set of investigators, many of whom were in the TCGA. And GDAN is I don't know the best term, but we work at the behest of NCI to do NCI things. And one of them was a set of projects that had been ordered for generating new models of cancer, the HCMI project models. And specifically, we have a very large number of cell lines. Maybe you might be familiar with the Cancer Cell Line Encyclopedia, which is a shareable resource in principle. But we don't necessarily have as many models for as many types of things as we want. They don't necessarily carry all the genetic drivers. Few of them have any clinical information attached to those cell line models. And many of them are not, like everyone's favorite breast cancer cell line, MB231, is a terrible breast cancer. I don't know what the hell it is. It looks nothing like any other breast cancer ever. So having faithful models that are shareable and can be recapitulated. And so we've got this enormous TCGA repertoire, ICGC repertoire of data, but we don't necessarily have models to do experimentation on. So HCMI set out to build these new models so that we could do this. And launched in 2016, more than about a dozen different centers contributed. Most of the models came from the Broad, Sanger, Hub, and Cold Spring Harbor, but there are other participants as well. And they exist either as organoid cell lines or neurospheres, so a variety of different types. And we have molecular characterization on all of these, as well as clinical data on all of these. So here's an overview slide of what's been done. And the goal was to be able to share models of a variety of different cancers that have been characterized and are faithful to the original tumor. And if they're not, we know it's different between them and have clinical information about the individual that they came from. So in total, we have 665 models that are available from ATCC today. They came from 637 different individuals. The largest fraction of them are from colorectal, but there are also substantial numbers of pancreatic, esophageal, a large number of GBMs, and you can read them as well as I can, as well as clinical information about how those patients were treated, both before and after donating tissue. We assessed the genetic faithfulness of these models by first comparing whether or not the mutations present in the model were also present in the tumor. We don't have tumor for every pair. We have 400 and change that we have paired tumor and model for. But you can see for the vast majority of these tumors, they are faithful. They carry at least 80% of the mutations in common. There is some subclonal graft growth. Some of them have evolved considerably. You can exclude those if you like. At the level of point mutations, they're extremely concordant. At the level of loss of heterozygosity, just asking about broad chromosome structure, they're extremely concordant. Basically, they're essentially perfect, in my opinion. When you look at other metrics, if you ask what mutagenetic processes were active, if you're worried that some cell model-based process might have created new mutations and being in culture is weird, we don't see any activity of new mutagenic processes active. There's very high correlation in the cosine correlation between the mutational signatures present in the models as well as the tumor that they're derived. Sometimes we see differences in the SNVs, but the mutational processes are the same, indicating that they really are they're independent subclones potentially. When we look at tumor ploidy, the models are slightly more likely to have undergone a whole genome duplication than their parent tumor. So perhaps we're selecting some subclones out, but very high correlation between tumor ploidy of the models and the tumors. And when you look at this, more than 90% of these models are faithful representatives of the tumors that they come from. So in terms of genetics, they're great. In terms of epigenetics and transcriptomics, they're also really faithful. So just going through asking, does the DNA methylation profile or does the transcriptional gene expression profile match? They do. You can see that these are highly correlated. More than, again, 90% of them. And these models are all commercially available. They're not cheap. but they are available. And so I don't know if there are bulk deals. I did get a UC-wide deal on some of them, so there might be a UC-wide way we can share some of them because they are expensive to grow and maintain. We've ordered about 20 of them from pancreas and colorectal. So if someone has a specific interest, I'd be happy to collaborate for the ones I've got ordered. All right, change of topic. That was all about cancer genomics of tumors and how we might go forward. I'm also really excited about populations. And the reason for that is, obviously, we are all affected by cancer. I've been affected by cancer in my family. I'm sure that many of you have as well. It's horrible. And there is a lot of underlying risk out there that is genetic in nature. So I started a project when I was at OHSU with Jackie Shannon, who's the head of population engaged research at OHSU, to see if we could engage the population of Oregon in a research set of projects. And our idea was that we wanted to, OHSU is a medium-sized medical school, but it does not have a very good patient base. And so going through the patient network didn't make sense to us. And so we went directly to the population. And our idea was we would identify scientific questions that would be appealing to members of the community. So we call that as a driver project. And we were going to be app-based, so all internet-based collection, very little in-person stuff. And we really wanted to make sure it was mutual information flow. So if we got something of value from a participant, we returned something of value to the participant. And the one I proposed that we start with was based on this Alaska Award piece that Mary Claire King wrote, which was pretty much an excoriation of the genomics community for not testing everyone for HBOC. And she lays out her rationale. This piece is published in 2014, and we are still not testing people for HBOC. And if you actually look, you can make basically exactly the same statement for Lynch syndrome. So basically, we know that there are huge numbers of people out there, on the order of 1 million US residents, have either hereditary breast and ovarian cancer syndrome, meaning that they have a very high likelihood of developing breast ovarian cancer syndrome if they're a woman, prostate cancer if they're a man and they have BRCA2, or in the case of Lynch syndrome, colorectal cancer. Something like 1 million people, probably someone in this room, right? And only about 25% of them are estimated to know it. And the reasons for that are covered in that Mary Claire King piece. But it's not anything that we as scientists would think is a reasonable reason why you're not, why we don't know. Like if you're an MD or like, like public health, maybe you would think they're reasonable, but like as a scientist, they're just like bullshit, right? So we said, let's do something about it. Let's just see if we can test everyone. And we looked at it, and the biggest single problem is that the cost of tests, the tests are geared towards being incredibly sensitive and thus are extremely costly. And as a genomics guy who spent most of my time figuring out how to make things cheap, I was like, well, I know how to do that cheaper. It won't be quite as good, but it'll be a lot cheaper. So we engineered like 97% of the cost out and retained about 90% of the sensitivity. And then we started launching this and seeing if people want to participate. And so we actually got this funded as part of the Biden moonshot to enroll a clinical trial of 25,000 participants, 24,500 who would receive our intervention, 500 who learned that they had HBOC or Lynch because of regular standard of care. So cohort A is standard of care. They came into the clinic as they do. Cohort B is anyone with a prior cancer diagnosis. We know that if you have a cancer diagnosis, you're more likely to have HBOC or Lynch syndrome. And cohort C was open to anyone who wanted to participate. And we made a second key decision, which is we only return pathogenic and likely pathogenic variants. So if you've ever seen one of these reports, they have VUSs in them. The VUSs are a nightmare. Do not touch them. They're terrible. They're a terrible idea. They made sense 25 years ago. They make no sense today. And we launched this test, and we did it in a framework. We were evaluating whether or not this was effective. And effectiveness means, do people actually do something with the information? Because if you give people a bunch of information about themselves and no one actually does anything, that's not actually useful. The second thing is it sustainable? And that means, what is its costs versus its outputs? And is the medical system capable of handling this? And the second one is often considered a deal breaker in the space because of the way genetic counseling has historically worked. And we then wanted to compare our participants to in our three arms, A, B, and C, and then evaluate this. And so this is the overall trial design with an estimated rough estimated numbers from the cohort A of 500, cohort B, those that had a cancer diagnosis of 130, those in the general population had 60. So the positives are HBOC and Lynch individuals. We did this all through social media. We just basically ran Facebook ads, and we did it all app enrollment. So you just downloaded an app and clicked on a link, requested a kit. We did it all. We were going to do some in person, but once COVID hit, we switched immediately to the mail. And we could control, because it was Facebook, we could control how much we spent on ads. We could maintain, you know, like not get asked for 1,000 kits one day and 0 kits for the next month. But Facebook is ruthlessly efficient at cost containment. It very quickly figured out who was going to enroll and who was not. And so by the end, we were at under $2 to enroll a participant in the study, right? So like ridiculously cheap. Everything else costs more, including postage, and you know, I think our kit cost was like engineered down to about $2 as well. So in total, we did this all in a CLIA lab, the Night Diagnostic Lab. 24,000 negative results were returned. 1,191 individuals were screened as positive on our first test. The Night had never run a scale like this and was unwilling to do it without 2 tests. So we required a second validation sample. Not everyone returned that. So only about 90% of the participants who were screened actually returned the second sample, so they get a clinical test. And of those 1,020, we have a long list of genes we tested for. HBOC and Lynch were just two of the syndromes we screened for. We had 163 HBOC and 69 Lynch, so 226 trial eligible participants. And so now we have actual numbers here. We're just in the process of breaking it down into those who had cancer, those who did not have cancer, whether or not they took up an intervention, whether or not they told family members who had cascade. We have preliminary data that says that people totally bought into this. If you're willing to participate, you're likely to do something, is roughly what you expect, right? So that's a very strong bias. Like if you're not forcing someone to do something, they're likely to want to take advantage of the information you give them. And so our goal is at least that even if we don't make people do this, we at least offer it. It's cheap, it's easy, it's totally doable. And the 2 key things to do are to not return the VUSs. In fact, like as an apocryphal story, we had a news article about some woman who had testing and had been given a VUS and then had a hysterectomy and prophylactic mastectomies for a VUS for Lynch. Right, that hit the Oregon newspaper about the time we were rolling out. But personally, I had a second, my stepmother's niece called me and was all in a tither, and she has a VUS, and she had 2 gynecologists tell her she needed surgery. So like what we're doing now is awful. It needs to stop. And we have a clear and easy solution, which is just test everyone, but not return the damn VUSs. And it's incredibly efficient if you do it this way. We had 3 genetic counselors return more than 1,000 results, which is way more than they would normally do in a year, right? Normally they would screen thousands of participants and return maybe 100 results, right? So if you just return the positives, it's wickedly efficient. So all that is the big reason I moved to UCLA, because OHSU is not configured to do this well, and UCLA is. UCLA has 7 million patient encounters in its database. UCLA has hundreds of thousands of unique patient experiences each year that are return patients, and has launched something called Atlas. It stands for @LA, and has already enrolled more than 150,000 participants in a genetic study led by Dan Geschwin and Clara Lajenchere, and a very wide team. So Atlas is not being done CLIA. It's just being done as a research enterprise. It has a universal consent, central biobanking, and all the data become available to UCLA faculty eventually. I don't know about the rest of the UCs. You might have to play nice. And the goal is to create a learning healthcare system where we have clinical records and genetics, including both GWAS backbone and exomes available for learning. So we have a dashboard. Currently, there are 340,000 participants who've signed the consent. They have not necessarily, sorry, 340,000 have viewed the consent, 220,000 have opted in. And so we can keep track of who opts in versus opts out. And then we collect DNA as remnants for all these individuals later on. And so we're doing that. We keep track of race and ethnicity data on all those individuals as well as gender. Women are more interested in this than men. Men suck sometimes. And so like this is, just, I didn't show you the data for HOP, but like HOP is like almost 3/4 women because that's who self-enrolls, right? Also who uses Facebook. And our ages are slightly age shifted. And one important thing about this is that the UCLA system is actually geared toward individuals with health problems. So unlike some biobanks that enrolled healthy people and now are waiting for people to develop health problems, the people in the UCLA health system are already experiencing health problems because that's why they're showing up already. And so we can keep track of this information about where they are. We're relatively better than most databases on being not just white folks. And we've done 130,000 genotypes and more than, and our next tranche of exomes will come in and put us at around 130,000. We've done just under 90,000 exomes today. A large number of them have actionable findings, things that they should know about. Something like 2 to 3% of these individuals have findings they should know about. We're not that good at returning results yet. We need to improve that process. So as you might expect, LA is on the west side. You can see where our participants mostly come from the west side, but we cover pretty much all of LA County. And in terms of ancestry and self-reported race, we're about 70% European, higher than that in white, but do contain substantial numbers of underrepresented minorities. And we're now just in the process of mining those data to both confirm existing publications as well as new things. And so we've, for example, I'm just highlighting a couple things. And I'd be curious if anyone has an explanation for this. If you look at LDRL null variants, these are individuals with familial hypercholesterolemia. They are expected to have high levels of cholesterol. And if you look at the top bar, all individuals in black and European ancestry individuals in green have very high cholesterol, much higher than you expect. If you look at various different subgroups for missense mutations, so these missense mutations are called independent of population. The average missense mutation in the European population has less effect, even though there are more null alleles in the European population. So that missense rate is way below the rates of missense variants in other populations. And so I can't explain this. It's really quite surprising. I'd actually just seen a talk from Illumina on a behavior like this, and it's totally inconsistent with that talk. So I just got this data the other day. And so I'm still processing it. But I don't, missense mutations that are estimated to be null should be null, and they're not. So there's something funny going on here, and it's specific to European variants. This is looking at genes involved in Parkinson's, showing ancestry-specific effects. In this case, the indigenous American chromosomes have different effect sizes for specific common variants with known phenotypes. And so these are the kinds of things you can tease out. And this is I think a particularly complicated one. Ronnie Haas, who's in Paul Boutrous' group, has been looking at all the polygenic risk scores, and we have polygenic risk score calculations on everyone, but she's been looking at them subdivided by IBD classed ancestry, so fine grain ancestry rather than the bulk European, African ancestries. And she's been looking at them and showing the breast cancer risk, in this case, by ancestry, by fine grain ancestry. And so the Puerto Ricans, we don't have a ton of Puerto Ricans in UCLA's database, but for them, they have lower frequency of breast cancer than others. And she showed me this data on Armenians, and I was really surprised because there's no known Armenian founder variants in BRCA1 or 2. If you do this for Ashkenazi, you can see the very strong effect size for Ashkenazi because of the strong BRCA1 and 2 deleterious variants that are founder alleles there, very tight error bars, and they're heavily enriched for breast cancer risk. But these Armenian populations, and to a lesser extent the Lebanese populations, have more risk than you expect. And I said, I think they're just coming to UCLA for care, right? Like, because the our median population mostly lives in Glendale. It's close enough to get to Westwood, but not close enough to come here for regular care. And if that were true, you would expect that overall risk for breast cancer would be different in that population. And she ran the polygenic risk score calculator, excluding individuals with a breast cancer diagnosis, and they too have more risk. So it's possible that it's real. It's right on the edge of what you can detect. But when you have these very fine-grained ancestries, you can start to tease out very complicated gene-environment interactions in potentially interesting ways. And this is built off the polygenic risk score trained on European ancestry individuals. So they have higher European ancestry estimated polygenic risk score for breast cancer, and they appear to have substantially higher breast cancer risk. So there's something interesting going on, and I think we're going to, as we accumulate more and more data, we'll start teasing this out. Okay, TCGA and PCOG are enormous. I don't have room for 1,000 people on there. There's some of my favorite friends in the world on this slide. This is Josh Stewart from Santa Cruz, routinely hosts in the lower left, for those of you who know Josh. So we usually hang out on the beach with Josh. The Van Loo Lab and I and my team have been collaborating extensively, and so you saw the work from Toby today a lot, but lots of other collaborators there. And for the HOP team, Jackie Shannon was the co-PI in that project, and lots of terrific folks from OHSU, including the diagnostic lab that was willing to let us develop our own test and deploy it, right, even though they ran it for us. So with that, I'll take questions. Rick.
So you showed some data that shows that at least that some mutation signatures arise at different times in the evolution of the tumor. Do you also see in parallel the defects that would cause those signatures arise in an appropriate, shall we say, kinetics?
Yeah, no, it's a terrific question whether or not we can actually do the accounting to show the MLH1 mutation and show that it happened at the same time. I haven't seen, we have not tried to do that, but it's a great idea.
Probably for this method, probably.
Yeah, could be methylation of MLH1. We haven't, with our long reads, we might be able to pull this one off now. So.
So how many different? You showed only a couple of examples of four, but of course there are many more signatures. And so I guess my question is, how many different signatures are you able to see sort of arising at specific times in different tumors, and does that imply a whole diversity of defects that then arising that then drive those signatures, even though you may not have seen correlate?
If I understand your question correctly, you're asking about in tumor 1.
Yeah.
How many mutation signatures do we estimate to be active in tumor 1? Is it, and when do they change and arise? Is that roughly what your question is?
Part of it. And then of course you have a larger set of tumors.
Yeah. So for the first part of the question, on an individual tumor basis, I am not aware of tumors that have very large numbers of signatures being active. 3, 4, 5 is pretty common. Getting beyond that when you deconvolve the signatures gets very hard unless they, although with the timing tricks, you might be able to segment the timing space in a way that would help you deconvolve them better. And potentially, I think we could talk to Lidmill about improving on that. But I think you're going to have trouble going to 20 signatures. And I'm not familiar with any evidence that would suggest that lots of tumors have lots of processes active. That's, you don't normally need, once you've got a mutagenic phenotype, you don't need another one. So there's not a selective advantage in engaging a second one typically. And I would think that because we can see treatment effects fairly clearly, it suggests that when you do have selection if there was an underlying tail of mutations of different signatures that happened late, they would have pulled up clonality post exposure, right? Because to see a mutation after treatment, you have to reduce the population down to clonal populations again. Right? So I think probably not that there would be 10, but it could be more than we can see. Yeah.
Maybe we do. Along with that question, have you or anyone else ever tried writing a I mean, people love writing new cancer gene callers. You can imagine based on what, this dynamic effect, if the signature background is changing between early and late, you might discover new cancer genes and turn some of those VUSs into, likely variants, or either at the variant or the gene level by, writing a mute-sig dynamo or a onco FM dynamo, dynamic onco FM.
Yeah, we have never written our own, so we've not. Gaddy works in this space pretty well, and I did just have dinner with Nuria the other day, so I'm not familiar with either of them.
I don't think they've done this, right?
I don't think so, no. But yes, I think it's totally plausible.
Nice talk, Paul. I have a question about the GRITIC paper. We just did this in Cancer Genomics Journal Club here. And one of the questions that came up is, you said about a third of cancers have this non-parsimonious evolution. Does that greatly affect the timelines that you guys published in PCOG for those tumors? Did you check?
We haven't redone the entire models in the 2020 paper. That was a lot of work.
Yeah.
And so we haven't recapitulated all of that work backward. I could get Toby to tell you what the estimate of how much improvement there would be.
Yeah.
It's more than 10%. It's not double.
And is it enriched in any certain tumors that process happens, or? I guess you didn't check them all.
I mean, a priori, it should be enriched in those that have a preference for whole genome duplication, which tend to be the hardest ones to do this on. So I would, things like ovarian, pancreas, quite frankly, the ones that we care about for the most, other than GBM, that have whole genome duplication should be the most impacted by this kind of thing. So, yeah. But Toby is definitely the one to ask.
Cool, thanks.
Any other questions?
All right, you're going to let me get away with all my controversial stuff at the end. Thank you.